Howdy, everyone. You're listening to Blue Jeans and Boots, where genetics grad students talk about how to navigate a PhD program while trying to stay sane. We also invite faculty from our interdisciplinary program here at Texas A&M onto the show and pick their brains about their research, what it's like being a principal investigator, and their journey to become a professor here at A&M. In this episode, we'll be talking to Dr. Matt Moulton. Dr. Moulton is an associate professor in the Department of Biology here at A&M. He received his bachelor's at Brigham Young University and his PhD at the University of Utah. His lab focuses on the molecular mechanisms of genes associated with neurological disease, especially Alzheimer's disease, by utilizing the genetic model organism Drosophila melanogaster. My name is Veronika, and I'll be your host today. Thank you for coming on our podcast.
Thanks for having me. It's a pleasure to be here.
Okay. So we're just going to kind of dive straight into it. So how did you decide what you wanted to study when you were in school, and how has that affected the research that you do today?
I think that it's been kind of a journey that I would not have predicted if you would have asked me 10 or 20 years ago what I'd be doing. But I did know that I wanted to study genetics. And so I thought probably the best place to do that is at a university. And so my goal was always to get a PhD and hopefully become a professor one day. So that was sort of my start into future career plans. I was really fortunate to be at a high school where I could take internship credit as part of my coursework. And so I was able to go to a local college and then a university for part of my senior year and get my hands dirty, so to speak, in the field of genetics. And so I got to learn about molecular cloning and about using model organisms for genetics research, and that really sort of sparked my initial interest in wanting to do genetics. And from then on, I was sort of dead set on it. Yes, exactly.
So you had all this exposure when you were in high school, right? That's quite early to be exposed to like molecular cloning. Yes, honestly.
Yeah. So it was pretty unique, and I feel really privileged to have been a part of that, and that helped, as I mentioned, solidify my interest in genetics. And so kept going in college. Tried to get in a lab, my first semester there, was able to start working in an insect systematics lab where we were using DNA to reconstruct phylogenetic trees for diverse groups of insects. And so I got to learn a lot of bench techniques and PCR and sequencing and things like that. And, the usual, right? And sort of ballooned from there.
Mm-hmm. So do you think that the research that you did in your undergrad studies also affected your PhD studies then?
Absolutely, yeah. So I think one of the best things that my undergraduate research experience provided me was that research experience and being able to be, learn tools and techniques that I could put on my CV. I was also able to be part of some publications as an undergraduate, which, isn't super common. And so I think that really helped build my application for grad school and allowed me to get into the school that I wanted to. And then continued, sort of in that line of genetics, wanting to do something genetics-based as a graduate student. And that was more in the field of developmental genetics, which I learned to really enjoy and grew to understand how an organism develops from a single cell to a very complex organism, and really solidified my love also for working in the fruit fly. So that was sort of my first foray into flies. And I really gained an appreciation for the types of experiments and that you can do in the fly, and the amount of genetic tools that are already available in the field that I could utilize for something that I was even more interested in doing, and that's sort of disease-relevant research.
So you started doing research in Drosophila already in your PhD studies,
Correct,
right?
yeah.
So can you talk a bit more about what your current lab focuses on?
Yeah, so right now we focus on using the fly as sort of a functional genetics tool. So one of the things that is challenging in human disease related research is understanding what a particular mutation does to a protein or cellular function. And so we're pretty good at finding mutations that may confer risk to disease, for example, Alzheimer's, which is a major focus in the lab. But then, really understanding how that mutation, or variant is what we call it, confers risk is less well understood. And so we utilize the fly in using a humanization strategy. So this is where we take out the fly gene that's equivalent to the human gene, and we put in the human gene in its place. And so we express that human gene in the same time and space that the fly gene is natively expressed. But now it's a human protein instead of a fly protein. And in the majority of cases, we can find that human protein functions properly, does its normal function in the fly, and the flies are totally fine and can tolerate that. But what that now allows us to do is to go in and now make single nucleotide variations in that gene, and then assess what happens when we make those mutations. So now we can say, okay, there's a disease risk mutation that for Alzheimer's disease or whatever disease we're interested in, put that into the fly and say, now what happens to that protein? Is it a loss of function, or does it have some sort of new function or altered function of some sort, and try to make conclusions on the human condition based on our studies in the humanized flies.
So would you say there are any any challenges when you want to translate the Drosophila research into the human disease research?
Yeah, this is a frequent question that we get, and this is not just me and my fly work. This is true for many or all of us in the fly community, is how translatable are these findings. And so it turns out that the fly biology, when we're talking really fundamentals of cell biology, the functions of fly cells are not that dissimilar from human cells. And this is true for neurons especially as well. We're studying this in the context of the brain. And so, our neurons have to, our neurons fire and have to metabolize, glucose, do all the same things that the fly neurons do. And so at a sort of fundamental level, there's not that many differences. And so when we're thinking in terms of what does a protein do in the cell, it's quite easily translatable from flies to humans. I think one of the powers of using this humanization approach is that it's immediately translatable because we're studying a human protein. And so, we're doing this in the fly because we want to do this in vivo. It's really important to us, we think, to understand the genetics of disease in a living, breathing, organism with neurons that are actually firing and glia that are supporting these neurons, et cetera. And so it's important to us to study these outside of like a cell culture model, perhaps, although there's certainly utility for those types of experiments as well. And so we want to do it in an organism, and the fly is a great organism in which to do that. Readily translatable, shortened lifespan, and these humanization strategies are fairly straightforward.
Yeah, I do research as well in a fruit fly lab, so I also have an appreciation for the Drosophila model. Definitely compared to some of my other cohort members who work, for example, with mice,
Right.
which takes a much longer time to actually get the mutated genes to show you some phenotypes that you're concerned about.
Yeah, exactly.
So what would be some long-term questions that your lab is trying to answer?
Yeah, so the genetics of a disease like Alzheimer's is incredibly complex, meaning that it's not one or two or even a handful of genes that are influencing disease risk. It's dozens or maybe hundreds. And so each one of those those genes is subject to variation at multiple locations. And so we definitely have our work cut out for us in terms of the number of genes that we would like to study and the number of variants within each of those genes. And so, as I mentioned, it can be fairly straightforward to find those, but then systematically going through one by one and assessing the function of those takes a significant amount of time. But that's a major aim of the lab, is to go through as many as we can, and then also utilize this for other diseases of maybe neurologically related or not. Anything that collaborators or even us as the lab is interested in, we can definitely move into and try to humanize and study the function of these variants.
So if you had unlimited time and resources, what kind of experiment or project would you work on?
So I think unlimited resources, we would definitely expand all the humanized lines that we're characterizing. There's just so many genes to be looking at and so many variants that would probably be priority number one. One of the things that I'm working on together with Dr. Alex Keene in the department is some drug screening, which is a bit of a challenge to do in a fly model, but he is very skilled at this. He and his lab members are incredibly skilled at this. And so we're able to start getting at least a small hold on some drugs that might improve lifespan and viability for some of the fly models that we're studying. And so this is really promising, but this is on a handful of drugs. And so given unlimited resources, definitely would love to expand this to many other types of compounds and maybe even compounds that have never really been characterized or understood yet. So I think in terms of therapies and helping patients, that could be really, really powerful.
Wonderful. What kind of mechanisms or genetic tools do you use when you're making these humanized flies? Is that how you call them?
Yep. So this utilizes an insertion of a transcription factor called Gal4, which I know you'll be familiar with, and the fly community is well aware of this. This is a transcription factor that's not native to the fly, but allows us to control expression of any transgene that has this Gal4 transcription factor binding site called UAS. And so basically we can put in the human gene that's downstream of the UAS sequence, and now we can control the expression of that gene by expressing Gal4. And so what we do is we insert the Gal4 into the fly gene. And what this does is it knocks out the fly gene, it eliminates its function, causes a truncated protein, which is typically a null, and then expresses Gal4 in its place. And so anywhere that fly gene is expressed, Gal4 will be expressed. And now if we add the human gene into that, we'll express the human gene in place of the fly gene at the same time and space.
So do you also, for example, do any like CRISPR work as well when it comes to like specific mutations then and like insertions?
Yeah, exactly. So in terms of putting in the human gene, we have what are called docking sites in the genome that are pretty standardized. And that's on purpose because we want the gene to go into a place that's not going to disrupt any other genes. So our trans genes sort of go there, and we have site-specific recombinase proteins that are stably expressed in the fly genome, and that's sort of standardized and straightforward. In terms of inserting the Gal4, we have to do that on a locus-specific sort of case-by-case basis. And so for those Gal4 insertions, we do use CRISPR to target it to a specific locus. And there we have, our construct has splice acceptor sites, and so we're actually targeting introns. So we're not hitting the coding region, just sticking it in an intron, allowing the Gal4 to be spliced in and then terminate any downstream protein coding or protein translation that would occur.
Good. Yeah. That sounds interesting. Okay. So I think we're going to move on from talking about the research more about your experience as a newer faculty at a university. So what has the first year of running your lab been like at Texas A&M?
It's been really great. We have some fantastic support here at this university. I think, even within my first month, I had people reaching out and saying, "Hey, we're here to help you write your grants, and we're here to help you, place orders and, with equipment and shipping and all the things that you need to do." There were people already in place who could help with all of those complicated things. I have definitely learned a lot. And much of that learning thankfully has been sort of smoothed out because of the people who are here to help, and also fellow faculty who have been on this journey before, and exactly what you're going through and can help. So I've been really grateful for my colleagues in the biology department, and then the university just broadly. We have great faculty and departments all over campus. It's an amazing place to be a part of such a richly diverse campus with just so many students, so many faculty. I feel like you can find answers to anything just right here in our own little bubble, so to speak.
That kind of reminds me of the reason I also joined Texas A&M, because during recruitment, because we all have to come and do interviews and kind of get to know the campus and the people, the reason I did choose to come here is because I felt that immediate support around me compared to like other places.
It's not hard to love A&M.
It's a unique place for sure. What do you think have been challenges that you had to overcome in the first year?
Yeah, I think one of the things I've found most challenging but also most rewarding is mentoring students. So as a new PI, you're sort of transitioning from doing the work to helping others succeed at doing the work. And so that's been a real challenge in terms of like I just need to learn how to be a better mentor. And so, that's something that I'm still learning about. And it's oftentimes different depending on who you're mentoring and things like that. And so there's lots of nuance there. So that has been certainly like a learning and growing experience, but it's something that I've found more like joy or reward in than I thought I ever would. So it's becoming, absolutely my favorite part of being a PI is working with the students in the lab and getting to help them learn new techniques and approaches and get successful at it and grow projects.
Would you say that you're hands-on still, like coming not that long ago from like a research position?
Yes, probably more than maybe others. But it's funny because, when I walk in the lab, things still go silent. Maybe this is true for you and your PIs too. Your PI walks in and everyone's like just a little bit more nervous.
Not my
okay,
lab.
good.
Yeah.
Good. Maybe I intimidate my students, right? So I try to be involved as much as I can, but also at the same time, like leaving as much as I can also, so that they have ample opportunities to try it on their own without me sort of breathing down their neck.
Yeah. Do you also have like your fly stocks and setting up crosses, or do you not do that anymore?
Yes. Yeah. I still have a little bit that I do, but most of it students, and I have a lab technician who helps with that as well. So they're pretty proficient at it. I think this is another sort of advantage of Texas A&M and the fly community is, our students are well supported as well. So they can come in and really hit the ground running pretty fast. I think it helps that flies are fairly easy to take care of as well. And so, all those things sort of help that my students can pick up rather quick.
Yeah.
And they're bright. We have really great students here.
Good. Yes, we do. What's something you wish you would know before starting your lab?
It's always hard to predict the future, of course. And so sometimes knowing like what experiments will I be doing in 5 years from now or something can be a challenge. And so, depending on where experiments will lead you determines what sort of equipment you'll need or supplies or even maybe how you'll train your students and things like that. And so that's kind of challenging. I don't know if that answers the question.
Yeah, it does.
Okay.
Just like something, I guess, that going into, being a new PI, having a mindset, maybe something that you should have known before, but you like got to learn it throughout the path.
Yeah, I think mentoring is something that could fit that. At least for me, most of my training thus far has just been like research,
Yeah.
and it's very, very much like you do the research, right? And now suddenly it's like, okay, help others do the research, and I think that's a very different. You need a very different set of skills. So if I could, transport someone else's ability to mentor into myself, like that probably would have helped, speed things up and made the transition smoother. But like I mentioned, it's still fun for me to learn. And I enjoy it. So.
I think that's something that they encourage us now to do as well. To, a lot of the PIs get a bunch of emails from undergrads wanting to join labs, wanting experience. So they're just like, oh, do you want 2 undergrads? Like, you basically have no choice, so you're gonna say yes. And you definitely start learning how to teach another person things that you at this point consider common knowledge, but like not to somebody who's obviously not in the Drosophila world.
Yeah, it teaches you a lot about what you really actually know and what you don't know.
Yeah. So would you say, I know you described a bit your journey in the beginning of this talk, but would you say that you always considered your career ending up in academia?
So the answer, I was thinking about this the other day, actually, and I think the answer is yes. And the reason for that is I think I was, and mistakenly, I was under the impression that that is where like you can do research, and that's what I wanted to do. And so I figured that you had to do that at an academic institution. And so it really wasn't until late grad school that I really gained a much better appreciation for all the different opportunities that are available for doing research. But having explored that, at least at some surface level, I was still sure that academia was the route I wanted to take. And so even though I never really deviated from that course, I recognize that I think it's really important for trainees to know the amount of diverse ways in which you can do research without necessarily being in an academic institution. I think they're the best, but but maybe not everyone agrees. And so I think it's helpful for students to at least be familiar with other options.
Yeah, I agree, because I feel like everybody usually asks if you want to be in academia or industry, and also like as somebody who's in the process of grad school, like people ask that all the time. But there are so many other options that you don't even think about because they're just not really spoken about that much. I guess.
Exactly. Yeah. When I look back at the cohort of just the people that entered grad school at the same time as me, I think maybe 2 of the couple dozen of us are actually professors.
Really?
Yeah. And most, I would say, are still in science, not all, but most are still in science, doing all sorts of interesting, different things that I don't think I really recognized as even possible paths. And sometimes that's the fun of science, is that it takes
you don't even know what you can do.
Yeah, right.
Yeah, this is great information. It's different talking to somebody who's been not that long ago and still like the learning process, and now like establishing a lab, because usually you think about a PI, oh, my PI had a lab for 15 years,
Right. Yes, yeah.
Yeah. So what kind of advice would you give for new advisors or grad students that are looking into continuing their path in academia?
Yeah. If you want to go into academia, you're at the right place. A&M is a fantastic place. I think for the professors that you'll meet here, most of us sort of wanted to do academia and stuck with academia. There are exceptions to that. And so it can be a little bit challenging for trainees who maybe don't want to do academia to find mentors. So I know that wasn't your question, but if you are not necessarily interested in academia, make sure, I think it would be really important for you to find, someone who can help guide you in those sorts of choices. But if you want to be in academia, you've got lots of great resources to be able to explore that. I do think, though, that it's important that we recognize that no two individuals, like if you were to find two professors here, you would be hard-pressed to find two professors here that have the same sort of academic trajectory or timeline or any of those things. It definitely seems like it's very variable. And so it all depends on, where you end up and what kind of projects you put on and, all these sorts of things that most of the time are not in our control. And so, being patient with that and being persistent with that, I think is always the key to finding success. Just keep at it. Grad school can be kind of, I use the phrase soul sucking. It's a horrible phrase. But because I've sort of seen that in like I said, many of my friends and colleagues going through grad school, many of them were just like, this is not what I want to do. I got to get out and do something different. And that's fine. There's, it's not going to be for everyone, but I think that you really have to be, sort of doggedly persistent in order to make it through. That doesn't mean you have to be the best researcher ever. It doesn't mean that your projects have to work perfectly, but it does mean that you just have to keep on trying.
Keep going. Yeah. I like how you brought up the point about how each professor or PI has a different path, because I think that's already something that I recognize now as being a grad student, that people have different timelines of committee meetings and prelims and papers, and so I feel like that's already something that you have to learn already during your path.
Yeah, for sure. And don't compare yourself to peers. That can be
what a sentence.
Hard too.
What a classic sentence. I know. Yeah, everyone says that, but it's hard.
It is hard.
It's very hard.
I've totally been there. Yeah.
So do you think you still deal with imposter syndrome?
100%.
Really?
Yeah. Like I wish I could look you in the face and tell you that I didn't, but no, it's like every day, like every faculty meeting, I'm like, am I really in the right place? My colleagues are brilliant and have, very successful labs, and, it's sort of starting out. Maybe it'll go away one day, but not today.
Not yet.
Not
Too
yet.
soon. Yeah. Do you think you have, especially in the biology department, other PIs that are also quite new as you are?
Yeah, I'm really fortunate that way too. The biology department has been really sort of prolific in their hiring as of late. And so, there were two professors hired at the same time as me and another person. I want to say a couple more the year before me, and sort of continuing on in the last 5+ years. And we continue to keep recruiting. We're hiring 2 more this year, etc.
That's great.
So I have a large cohort of new PIs.
Cohort of PIs.
Helpful. Yes, it's really great.
That's cool. I never thought about that. Okay, so we discussed research, we discussed mentoring. So what do you do in your free time to stay sane?
This is sort of a loaded question for me, because I have, my wife and I have 4 sons, and so I find myself not having free time.
That's your free time.
Most of the time, that is my free time. But I find that actually one of the most rewarding things in my life. So I won't go into that too much, but that definitely, I spend a lot of time just being a dad and playing and dropping off and picking up and things like that. But I actually really love it. I do also like to stay sane, to your question. Music is a big part of that for me. So I love listening to music. And that sort of keeps me grounded, helps me stay emotionally level. When things are bad, just even listening to a song can go a long way to sort of turn the mood around. So that helps me. I play the piano, and so I try to squeeze that in whenever I can. Yeah, that's kind of the big thing. Good. Being outside is always helpful. Sometimes that's hard in Texas.
Reconnecting with nature.
Yes. In the Texas summer, I just look outside. I don't instead of being outside, I imagine myself outside. But in the winter, it's great. It's nice to be able to be outdoors and enjoy.
I mean, you haven't been here a full summer.
I haven't been here full summer, but just prior to this, I was a postdoc in Houston.
Okay, so
I was there for a number of years, and so it doesn't feel too different. Maybe a little better, but
oh, really?
Maybe some days are a little less
questionable.
It's pretty breezy here in College Station, which helps. So.
I still think it's really hot. I was driving over, and my car said it was 100 Fahrenheit.
Yes.
It's quite hot today.
It is hot.
So how do you balance the demands of research, teaching, and mentoring? And what kind of strategies do you use to effectively manage your time and priorities?
Yeah, this is a super challenge, I think, for everybody, right? We all have a million things that we're juggling at all times. And you'd like to think that, things are going to settle down, as soon as this grant gets submitted or as soon as this paper's right. And the truth is, it never does, right? Something always sort of fills its place. And so, something just even simple for me is I have to calendar everything. So my calendar is just like full of to-dos and meetings and, my trainee schedules and things like that. It's kind of the only way that I can remember to do everything. And that's sort of the way that I try to find time for things. Something that was recommended to me was like, hey, not only schedule all those meetings and things, but schedule time for yourself. I'm going to write this paper during this block of time, right? And I can't schedule meetings during it because I'm writing this paper or I'm writing this grant or whatever. So that can be helpful. Otherwise, I think that sometimes you can't do it all. And that can feel sort of scary, at least for me. Like I would like to be able to do it all, but sometimes like I drop the ball, I forget something, and you just have to just try again. It's part of life. And maybe in that same vein, saying no to certain things is okay to do as well. For myself, I have a pretty hard stop at like 5 p.m. It's not always feasible, but as much as I can try, I'd say, I'm going home. I got to be with my family.
Yeah.
And, some of the other important things, the things that are important to me in my life as well, I got to make time for those. So, I can't always get out at 5, but I try to at least tell myself it's time to go home and be
prepare
done.
yourself.
Yeah, exactly. So I think, giving yourself ample time to be able to be who you are, whatever that is, in addition to doing great research in science, I think helps you be a better researcher and vice versa, a better human being. Because at the end of the day, it's not all about science. It's not all about research, although of course that's my favorite part, right? It's a big part, but it's about like who we're becoming and who we as a field, we as individuals, we as a community. In order to make all of that better, it starts I think, with us. And so, making time for ensuring that we are who we want to become is really important.
So how would you maybe describe like just a day in your lab for you?
A day in my lab often starts with collecting flies in the morning. So collecting virgin females, as the fly geneticists know. Yep. Usually that's the first thing you do in the morning. Typically, I'll try to meet with students. I try to meet with all my students at least once a week. Sort of have an open door policy. They can come whenever, but we have a sit down meeting where we can talk about updates on projects and things like that. So the day is usually sort of filled with some of those meetings. Sometimes I'm invited to be a guest on a podcast.
Yes.
That's not typical, but it does happen. Let's see. There's always lots of seminars to go to. My day is often filled with seminars. Most of my day is filled with writing, so sort of depending on what the next thing is, often grants, but also papers, sitting on committees, and so meeting with students in that regard. Let's see, what else?
Do you teach any classes?
I do teach classes. Well, I should say I will be teaching classes in the fall, starting in the fall. So I haven't had to teach yet.
So you were just like settling in so far. Yeah. Do you know what you're going to be teaching?
I'll be teaching a new course called The Biology of Neurodegenerative Disease. So we'll be learning about Parkinson's and Alzheimer's, ALS, MS, Huntington, and a few other less well-known degenerative diseases. So this is going to be a brand new class in the.
Is this an undergrad class?
For undergrads? Yeah.
Shame.
Although anyone's invited, you don't have to be enrolled.
But the prereqs don't really align that way. But that sounds great. Did you design that class?
I'm designing currently.
Present
tense. Designing the class. Yeah. So the hope was for the biology department to diversify some of their elective credits and bring in people who can teach things that are new, and so that was kind of the idea.
That's great. Okay. So what advice would you give to yourself before you started graduate school?
I think again it sort of comes down to like be patient and be persistent. It's easy, again, sort of comparing ourselves to other people to be like, oh, well, they already have their first paper, they already have this or that. But we're sort of running our own race, and so being patient, especially with ourselves, being patient with the experiments that we're doing, and also persisting. So much of what I have done in grad school and in postdoc, I find it just didn't work the first time, but that's because often it was something simple that I had to tweak, but you don't know until you really dig in and sort of figure it out and troubleshoot. And I find that fun. That's part of the fun that I think science is having the freedom and flexibility to do just that. But it does take a level of persistence or stubbornness, as the case may be.
Both.
Yeah,
hopefully.
both.
Yeah.
So sticking to it and letting things sort of run their course. Be patient with that.
I think that's great advice. Thanks. Thank you. All right, thank you so much for coming on this podcast.
It's been my pleasure.
It was very insightful, I think, because a lot of us are in the stage of building our careers, and you are somebody who just kind of wrapped that up and started a new chapter. So very insightful information.
Thanks. Thank you very much. You all have an exciting time ahead of you. Grad school is a great time to be, it's always great to be a grad student and enjoy the time that you have as a student. It gets better. I think.
He thinks.
But, that hopefully is motivational to keep going and keep doing what you're doing.
I just tell people that I'm a fake adult. So
yeah, I think we all are.
I'll take that.
You may not grow out of that.
I'll take that. Today's episode was hosted by Veronika and produced by Charles Mitchell. This podcast is brought to you by the Texas A&M University Interdisciplinary Graduate Program in Genetics and Genomics. If you have any questions about our program here at A&M, please feel free to email us at bluejeansandboots@gmail.com. You can also find us on Facebook, Instagram, YouTube, and X at Genetics Podcast. Thank you for listening. We hope you'll tune in next time. So.